Well, thanks everyone for attending today's citywide medical grand rounds. I'm Mike Farku, the Vice Chair of Research, and I'm representing Dr. Arnold Kumagai, who was unable to make it for the introduction. He's going to attend later. And so we're really pleased and honored to have our 2020 Canada Gardner-Whiteman Award laureate, Professor Guy Rouleau, join us. Dr. Rouleau is the director of the Montreal Neurological Institute, chair of the Department of Neurology and Neurosurgery, and director of the Department of Neurosciences at McGill. And he has made incredible contributions to our understanding of genes and their interaction with neurologic and psychiatric disorders, including autism, ALS, hereditary neuropathies, epilepsy, and schizophrenia. He's provided a better understanding of the molecular mechanisms that lead to these disease symptoms and has made contributions towards the discovery of new mutational mechanisms that could potentially lead to novel therapies. Dr. Rouleau has published over 900 peer-reviewed papers in the literature and has trained over 100 graduate students over the years. He's involved in something that's incredibly novel, known as the Tannenbaum Open Science Institute, which is pioneering a new way to doing research by adopting open science principles in order to accelerate discovery and benefit patients and society. Dr. Rouleau, we can learn a little bit about that sometime during this hour, but it's really a privilege to welcome you. The title of the talk is Molecular Genetics of Essential Tremor. Welcome, Dr. Rouleau.
Thank you very much for this kind introduction, and I'm very happy to be here today to present these rounds, which are being seen by many people. I found out that it was virtual about 5 minutes ago or 10 minutes ago, but that's fine. We're all used to it now in this post-COVID age. So I'm going to talk to you about a disease that I'm quite interested in, essential tremor. And I have a few disclaimers which are written here, but essentially there are no conflicts of interest or financial interest related to this talk. People in my lab also wish me to declare that I did none of the work that I'm going to be presenting. So the disease I'm going to talk about, essential tremor. It's one of the most common neurological disorders, and it's estimated the prevalence at all ages is almost 1%. And the prevalence increases with age, reaching 4.6% over 65 years of age. So it makes it by far the most common movement disorder in humans. It's characterized by a postural tremor that worsens with movement. So there's a small tremor, usually rapid, small amplitude, and it increases with use. So typically people if they're trying to drink their coffee or drink some soup, they spill as they are bringing it towards their mouth. It mainly affects the upper limbs, but can affect voice, the head, tongue, face, lower limbs, and trunk. And it can be severe to the point where there's a functional disability, even incapacitation. But I would say that would be a minority of individuals. What's important to know is that it's a syndrome. It's not a single disease. It's likely a lot of significant heterogeneity. And when you see a lot of patients with essential tremor, you note that they're not all the same. There are some subtle differences between the different individuals. Pathophysiology remains unknown, though there's some data that suggests a neurodegenerative process in the cerebellum. There's also possibly involvement of the cerebellar olivary circuit, but certainly the cerebellum seems to be the place where the action is. There are no effective treatments, but beta blockers and primidone does help in some patients. This is a typical test. You ask the patient to copy a spiral. They cannot rest their arms. They have to do it without resting their arm. And you can see the tremor is quite obvious. It's small and high amplitude, high frequency. So what about the genetics of essential tremor? We know it has a significant hereditary component because over 50% up to 70% of cases have either more than 2 immediate family members affected with the disease or 2 family members diagnosed before the age of 65 years of age. So And the concordance rates in ET are high for monozygotics, 93 to 77, and lower, but still significantly high for dizygotic. So using the classic formula, we arrive at a heritability of about 70%. Nothing is known about the environmental factors that can influence this disease. Now, if we go back many years, this in the 1990s, there were a number of genetic studies that found 3 genomic loci, looking at families where there are many affected individuals. But, oops. Okay, so all these loci identified by linkage, in fact, none have been replicated. No genes have been identified. And most people think that these were essentially false positive findings. So I think we can forget about those. Now, I started working on this disease quite a few years ago. We had focused on the French-Canadian founder population. We collected quite a lot of French-Canadian families and a lot of unrelated affected individuals. So linkage families were performed and really did not work. And you'll see in a minute why it didn't work. Essentially, it's diagnostic uncertainties because everybody has a bit of a tremor, physiological tremor, and the border between physiological and essential tremor is not always easy to establish. So to overcome the diagnostic barriers, we decided to do exome sequencing. This is one of the first exome sequencing, if not the first exome sequencing study my lab did well over 10 years ago. And we did sequencing only of those who had definite essential tremor and an age of onset of under 40. So more likely to be true essential tremor patients and familial. This is a family, you don't have to look at the details, big family, a lot of affected individuals. It was a site visit that we did. Interestingly, I did it with a neurology resident at the time who now is the Dean of the Faculty of Medicine at the University of Montreal. So this family was collected a long time ago. So we only use the ones who are definitely affected. And this is the result of the exome analysis. So if you look at the bottom of the slide, you see there was only one gene that had a mutation in all the affected individuals, definitely affected individuals. The mutation was in the FUS gene. Now, this was quite surprising to us because a few years earlier, we and others had shown that mutations in the FUS gene cause amyotrophic lateral sclerosis, familial ALS. So we were a little surprised, but that's what the data showed. So if we look a little bit more in detail, you can see that those who have definitive ALS have, all of them, 100% have the mutation. Those with probable, not ALS, sorry, essential tremor. Those with probable essential tremor, also 100% were mutation carriers, but those with possible, so those where we had the least diagnostic certainty, only 54% had the mutation. So you can see that if you do linkage, you tend to try to use everybody to get enough power. And if you'd use linkage using these possibly affected individuals, you would get false, you would not find linkage. At the clinically unaffected, there was none. And what was interesting, it was a nonsense mutation in the nuclear export signal of the gene. So this mutation, which is this nonsense mutation, has not been reported to date. So it's extremely, extremely rare. It was not detected in ethically matched controls. And it's only the third nonsense mutation ever reported in the FUS gene. The other two, R495X and Q519X, cause amyotrophic lateral sclerosis. And one of these actually my lab described. So we started thinking, what is happening? So if we know that in ALS, the mutations are clustered in the nuclear localization signal at the C terminus. And this is an area that has prion-like sequence and prion-like properties. So these mutations, we think, are an aberrant gain of function or toxic property. And if you look at the ALS literature, there were no mutations in any ALS family or cases in the nuclear export signal where this stop codon is. In ET, the stop codon is in the middle of the protein. So usually if there's a stop codon in the middle of the protein, you have nonsense mediated decay, which leads to the degradation of the mRNA and no protein. In contrast, the mutations in ALS are in the C terminus, the two protein truncating mutations I mentioned. And mutations that are very far in the C terminus often escape nonsense mediated decay. So our hypothesis was that essential tremor results from a loss of function, while ALS results from a gain of function. This predicts some things. It predicts that ALS truncating mutations, but not ET truncating mutations, should lead to a protein. And as I mentioned a minute ago, the ET truncating mutation we would predict would be a target of the nonsense mediated decay system. So this is a complex slide, but I think if you just look at panel C here, this is allele-specific expression of the two different alleles that we see in the ET cases. So in the ET cases, there's one allele that's normal, and one allele has the truncating mutation. And so you see here on the left, non-treated, we have none of the mutated allele, no RNA of the mutated allele, because this is looking at RNA expression, and you have a good quantity of RNA from the normal allele. If you treat with puromycin, which inhibits the nonsense-mediated decay, it doesn't inhibit perfectly, but it does reduce the nonsense-mediated decay, you see some of the RNA with the ET protein truncating mutation starts to appear, indicating that nonsense-mediated decay does occur and that this is a pure loss of function with no protein that will be produced. But if you look at panel A, this is the ALS case that has a protein truncating mutation, and this is ET. So you can see in the ALS case, you don't really have much effect of puromycin. You don't get an increase in the amount of RNA with puromycin, but in the ET case, you do. So that confirms our hypothesis. So conclusions of the exome study. So we identified a nonsense FUS mutation that segregates with essential tremor. There is no ALS in this large family, which of course, you know, the penetrance of FUS ALS mutations is virtually 100%. So it's surprising that there are no ALS cases, or maybe not surprising, it's probably telling. And that mutant ET RNA undergoes degradation by nonsense-mediated decay, while the ALS RNA does not. So it suggests gain of function in ALS, loss of function in ET. And unfortunately, this has not been replicated by anybody else other than a Chinese study that found an association with a FUS variant in their cases. So it's obviously extremely, extremely rare. So what is a genetic architecture of essential tremor? Well, there are several rare genetic variants in different genes can predispose to ET. What about common variants with low penetrance? These you test by genome-wide association, something you've certainly heard about many, many times. This may be a slide that you've seen many times, but essentially what we're looking for is common variants with very low, common variants with low effect size. In other words, low penetrance common variants. So there were some small-scale genome-wide association studies that were done. I won't talk about these because we in 2015 published a much larger ET-GWAS, found 3 significant loci, which I'm just going to gloss over because there's a lot more. There's a follow-up. We found some loci. Then we organized a second GWAS, and this is a very long list of collaborating centers, and we published in 2022 the largest GWAS to date for essential tremor. Essentially, there were 7,100 cases, 475,000 controls, many different centers. We had also participation of 23andMe, who actually contributed quite a large number of cases, and the UK Biobank, which contributed almost no cases at all. So this is the result. So we identified 5 genome-wide significant loci. This one is by far the strongest, and I'm going to talk more about it in a minute. These are the details. So essentially 10 to the minus 14, 10 to the minus 8 is the span. These are the different loci and chromosomes. And interestingly, the strongest association on chromosome 21 is an intronic region of the base 2 gene, which is associated with myelin abnormalities in the inferior cerebellar peduncle. So we're talking about, again, the cerebellum. This one here, this one number 46, was replicated as well from the previous GWAS. So we started to try to see how can we validate this and get a better understanding of what's going on. So we thought, let's look at these genomic risk loci, these 5, and see where are they highly expressed. And what we see is that they're highly expressed in the cerebellum. They're specific for the brain, but highest expression is cerebellum. Certainly very consistent with these being true loci for susceptibility to essential tremor. Then we did a transcriptome-wide association study, which is basically where you combine GWAS with RNA sequencing data, where you try to predict the effect of common variants on expression. And this is a bit of a complicated slide, but I think what's important to note is we have down here in red one of the most significant, or I think it was the most significant outlier was in the base 2 gene. So it's actually predicted to be downregulated in ET patients, meaning that this variant that we found that is highly associated is associated with reduced expression of base 2. So that certainly adds more evidence that base 2 is one of these predisposing alleles. And next, we had access to brains from patients with essential tremor who had died, individuals who had died, and compared them to controls. As you can see, we confirm that base 2 expression is reduced across all brain regions. So essentially, there are 3 lines of evidence that seem to suggest that base 2 is a good candidate. So we are doing follow-up studies, and I don't really have much data to show you. In fact, I have no data to show you, except to say that we're using CRISPR-RI in human-induced pluripotent stem cells to knock down base 2, to reduce the expression of base 2, down-regulated, and we're going to look to see the transcriptional effects and if there are any phenotypic effects of this gene. So this is a molecular follow-up work in progress. So the takeaways: largest ET-GWAS to date, identified the first genome-wide significant loci, reiterates the implication of the cerebellum in the pathogenesis of ET, and that base 2 is a good candidate down-regulated in this disease. What about rare alleles causing Mendelian disease? I did talk about FUS as one, which is a very rare, highly penetrant, but we wanted to see if we could identify certain others. So there we used a method called MIP capture. It's just a relatively rapid, relatively inexpensive method to do deep sequencing of genes. So we used this method. We had discovery cohort of almost 800 cases and a replication cohort of almost 900 cases and an appropriate number of controls. So what genes did we want to look at? We thought that we would look at genes that were associated with diseases that have been linked with essential tremor. And migraine there are a number of studies that have linked migraine with essential tremor. So we asked the question, is there shared genetic factors that may underlie the comorbidity of ET and migraine? So these are 6 genes that have been implicated in migraine. So we looked at these 6 genes. Some are really very big genes, like the calcium channel and the sodium channel. And so this is the result. So we had a suggestive excess of rare alleles in this calcium A1A gene, 30 versus 19. It is significant, but not highly significant. And what's interesting though is that we then again, I'm wondering if that's the other slide, but anyhow, we had seen this gene, the CACN A1A, in another study where we did a multi-omic study and looked at expression of genes in brains of patients with essential tremor. This is a paper Liao et al. 2019. I'm not talking about this paper. I'm just mentioning that we had another study that pointed to this gene. So this would be two studies that seem to suggest this gene. And it's just to say we did a replication cohort, and you can see the replication cohort here is also significant, and that the combined cohort is even more significant. So given that we've seen this gene using two different approaches, one genetic and one multi-omic, it does suggest that this is a reasonable candidate gene that could lead to this disease. What is this gene? Interestingly, it's codes for voltage-gated calcium channel, and it's most abundant in the Purkinje cells being, of course, the sole output neuron of the cerebellum. So again, pointing to the cerebellum. Then we looked at all the different animal models, and we found one, which is this KCNN2 gene, which is a calcium-activated potassium channel. There are a lot of potassium channels. For those who don't know about potassium channels, there's almost 100 of them. But this one, KCNN2, when mutated in a mouse model, led to an autosomal dominant whole body tremor. So we screened this gene, and what we found was, if you look at this, just this one, protein altering rare alleles, we had 7 in the cases, 1 in the controls, very weakly positive. So I'd say, you know, one line of evidence with weak data supporting involvement of this gene in the disease. It's an interesting because 2 of the genes are actually in this calmodulin binding domain here of this protein, which is a regulatory region. That could be a place, or it could be interesting to follow these up. But at this point, we're going to let this one rest and let somebody else follow it up because we have others to work on. Now, we've been talking about sequencing studies. You know, CNVs are another kind of mutation that one does not easily detect using sequencing studies. And so we decided to look at CNVs in our data set. And CNVs you can look at when you do SNP arrays for GWAS. And so we were able to look at a lot of our cases, 2,100 cases and 10,000 controls. And we called the CNVs using, you know, classic methods. And what we were looking for were rare CNVs that were associated with the disease. What we found, interestingly, is another calcium channel, this one, the A1H. And we found, as you can see here, it is only in the cases. So in the cases, there were 9 duplications. In the controls, there were 0. And we had almost 10,000 controls versus 1,400 cases. This is highly significant. And we validated that these are indeed duplications using another method. So this is actually quite interesting because it's another voltage-gated calcium channel similar to the A1A. This disease has been shown to lead to, if you have more severe mutations, to childhood onset cerebellar atrophy. So again, the cerebellum, and in some individuals, epilepsy. So interesting evidence. So what's our conclusion about the rare variants? Well, genes are likely to be involved. The 1H I just talked about, FUS, and the 1A genes, the calcium channels. And genes possibly involved are the KCNN2s. And we need to have more genetic studies, which probably will have to be done by somebody else, because we've used most of our cases, or functional studies, which we have done for FUS and are underway for the 2 calcium channels. we used another approach, which is looking at transcriptomics from drug treatment. So I had mentioned at the beginning that there are two drugs, propranolol and primidone, which are effective or partly effective in a fraction of cases with ET. Mechanism of action is not very well known, but we know that a lot of the actions of the drugs can be through changing transcriptomics, transcriptome. So we decided we wanted to look at that. So we thought, okay, we're looking at two different drugs, so maybe the transcriptomic targets of the two drugs, independent studies, converge on relevant ET genes. This is the experiment. So we used two kinds of cells, the NPCs and DAOY cells, treat with the medication, do RNA-seq, and then look at differentially expressed genes and then cross them to see which are differentially expressed in the same direction in both with both drugs. So what we found is that these genes that are differentially expressed converge on axon guidance and neuron morphology. So that certainly is compatible with what we think might be going on in the disease. But here, you're going to see we have a lot of you can see we have a lot of approaches, and what we like to do is try to intersect the approaches. And so one of these is this SHF gene, which was part of this Liao et al. Paper that I've not described, but there were two significant hits in this paper. One was the calcium 1A1, and the other one was the SHF gene. And so we looked at the SHF gene, and we found that it was one of the genes that is affected by propranolol. So we took some lymphoblast cell lines from patients with ET, and we treated them with propranolol. And as you can see here, we have a significant increase in the expression of this gene, which was predicted to be downregulated in ET. So let me just repeat. Did a study in ET brains, found that this was one of two genes that was downregulated in ET brains, and this drug, propranolol, leads to an increased expression of this, and we find this specifically in cells from ET patients. So certainly suggesting that the SHF gene may also be important. So we were interested in knowing what cells are involved with ET. I've been talking to you about the cerebellum. Certainly there's no consensus what part of the cerebellum, which cells in the cerebellum, but people love Purkinje cells. It's one of the favorite cells of neurobiologists, and so people say it must be the Purkinje cells. So we thought, okay, maybe could cells with high expression of ET genes also those that are targeted by ET drugs? So basically we want to combine the expression and the ET drug effects. So we looked for enrichment of expression in ET genes in different cell types in the cerebellum, and then we looked at the genes targeted by ET drugs. So ET-related genes, they're enriched actually in astrocytes and oligodendrocytes, so not the Purkinje cells. It seems to suggest that the cerebellar astrocytes and oligodendrocytes might be these cells that are important in the pathogenesis of essential tremor. Then we looked at the E3 drug targets, and we find again that they're enriched in astrocytes and oligodendrocytes, again pointing to astrocytes and oligodendrocytes. So our conclusion, basically, these propranolol and primidone genes, affect genes converge on axon guidance, and the ET genes and ET targets have enriched expression in cerebellar astrocytes and oligodendrocytes, and that these might be important cells in the pathogenesis of essential tremor. This is my last study. So we looked at the ET single-nucleus RNA sequencing. So we have 16 ET brains and 16 control brains, and we looked at the cerebellar cortex. We now have access to many more brains, and so this study is going to be greatly expanded. We use split pool ligation-based transcriptome. Some people who know this method, a lot of people use 10x, but this one is cheaper well, cheaper, basically cheaper. And we sequence close to 50,000 cerebellar cells. This is what it looks like. This is a very typical plot that you see. It's basically a principal component analysis. And you can see that the most abundant cells by far are the granule cells. And we are in the process of trying to determine the subtypes of granule cells and are there differences in the subtypes of granule cells. Here, very small here are the Purkinje cells. So there are not a lot of them, but they are, of course, very, very important. Here we have the astrocytes and the Bergmann glia and the oligodendrocytes, okay? And then other kinds of cells. Basically, we find all the major cell types we can identify in the cerebellum. So if we look at what's going on in some of the glial cells, we see that there's evidence of astrocyte activation in the Bergmann glia cells. And so that certainly is indicative that there's something going on at the level of the Bergmann glia. And what are the Bergmann glia? So this is a cartoon of the cerebellum. You can see in red here the very important Purkinje cells that sends out its axon to the cerebellar nuclei, and that's the output. The sole output is through the Purkinje cells, but the Purkinje cells are influenced by many cells, stellate cells, basket cells. But the Bergmann glia essentially are specialized to maintain and regulate Purkinje cells homeostasis. So certainly any changes in Bergmann glia you would expect would have some changes in Purkinje cell function. So what are the conclusions of our single-nuclei RNA sequencing? Well, again, this is the first study that does that in ET brains. Preliminary analysis suggests non-neuronal cells, especially Bergmann glia, are the main affected site to that cell type. And there is evidence of Bergmann gliosis. Now, is it something that is a consequence of the neural dysfunction, or is it inducing neural dysfunction? This we don't know. The old chicken and egg story. So we're looking to increase the number of sequenced nuclei and of patients. So my overall conclusion, we've identified a number of genes that predispose to essential tremor. Most confer small risk with the exception of FUS. The cerebellum and possibly astrocytes and oligodendrocytes may be the critical sites for disease pathogenesis. Our pharmacological approach, I think, is promising to add to our ability to interpret the different kinds of data that we generate. So future directions, well, two directions. One is we want to increase the genetic power by obtaining more cases of genome-wide association, and there are some pockets and some groups that have some of these, plus 23andMe has continued to grow their collection. So I think this is realistic. And we've started a consortium, a worldwide consortium for GWAS, exome and genome sequencing, so that we can pool all the data of all the different labs who are doing this kind of work. Now, this being said, our lab has by far the largest GWAS and by far the largest exome and the largest genome sequencing data, but we combine with other labs, I think it'll be very fruitful. And we want to validate the strongest candidate genes using different methods. My lab has been using a lot of iPSC experiments, so I can induce pluripotent stem cells differentiated into cerebellum, maybe in cerebellar organoids, is something we're looking into. So, as I mentioned at the beginning, I really didn't do any of the work. These are the students, Carl, Gabriel, Charles-Etienne, Miranda, who did a lot of the work. Danielle, Hélène, and Patrick are kind of the supervisors and run the lab. Sylvain Chunard is a very important collaborator from the University of Montreal, has been with us since the beginning of this study. So I'm open to questions at this point, and I'll stop sharing my screen.
Thank you very much, Professor Rouleau. That was a tour de force and an elegant lecture about how to develop these strategies of genomics and moving forward to potential therapeutic interventions. We're going to open it up for questions. I think I have a question here from the chat. Let me just get it for you. This is from Dr. Glabman. Is there a relationship between the genes identified and disease expression in terms of sex, age, and severity of the tremor?
That's a good question. So we know that for FUS, the FUS family, it's an extremely severe tremor. So I would say that there is an association between the genes identified and the severity of the disease. But there are not that many highly penetrant genes that have been definitely implicated in the disease. So certainly the data seems to indicate that there is that. The severity does increase with age. There's no sex difference as far as we know. And there is an age of onset factor. And certainly in the literature, it seems to say that if the onset of tremor is before 40 years of age, it tends to be more genetic. And if it's after 40 years of age, it tends not to be genetic. We did look at that specifically and found no evidence for that whatsoever in our genome-wide association studies. But I tell you that the FUS family, that I remember examining them, they had very young onset, but not childhood, but in the teens, of a tremor that could be quite severe. Yeah.
Wonderful.
I think one thing that I didn't mention that maybe this question kind of brings to mind. I mean, I did not present all our work. I can tell you right now, there's a lot of stuff that wasn't presented. One of the things I didn't present is that if you go in the literature, there are a lot of possible essential tremor genes. And in our MIP study, doing this large scale, like almost 1,500 cases and a few thousand controls, we tested all the candidate genes that are in the literature, and they're all negative. So all the candidate genes that have been published, of which maybe there's 5 or 6, we found no evidence whatsoever to support that they are real genes.
I'm going to ask on Sandra Black has a question, professor of neurology.
Well, first of all, Guy, that was fabulous. Just a wonderful detective story, identifying actual cell types in the cerebellum. It's just wonderful to see this work. But I have a question. Having figured this out, as you know, currently approved treatment for severe essential tremor, kind of 2 wrongs making a right, is with focused ultrasound. You can do it also in other ways, but focused ultrasound is the least invasive. You knock out some, I guess, cell groups within the nucleus intermedius of the thalamus, and it can cure severe essential tremor, like on the opposite side. Do you have can you sort of explain what may be going on there between the cerebellum and, I always sort of thought it was basal ganglia that was involved, and it sounds like the cerebellum is really the seat of this, but any thoughts on that? Because
well, you're absolutely right that ablation of the subthalamic nucleus has been shown by neurosurgical ablation in the past, and now with focused ultrasound to help alleviate the tremor. And I must confess that we have not figured out how that works at all. I can say that I have quite a few of my patients have had that, and it works really well, but I wouldn't say it cures them. Usually it works very well at the beginning, and they have maybe 80–90% improvement, and then the tremor comes back, some it only comes back a bit, some it comes back quite a lot, but anyhow, they're better, and we only do it in people who have so severe tremor that they can't function. And the other problem is you can only do one side. No neurosurgeon is going to want to do both sides, so you got to do the correct side, or the one that the patient needs the most. But the circuitry, I do not know how it fits into this cerebellar circuitry that we're looking at. That's a very good question.
thank you.
Dr. Rouleau, I wanted to ask you about the Tannenbaum Institute, this whole model, because it seems like it's an accelerated way of advancing science, and you've done, I think this is a demonstration of that. Tell us about this model that you've developed.
Yeah, so about 2 hours ago, I deleted those slides that I had at the end of the talk because I thought I wouldn't have time. So I can just say in a nutshell, the notion is that certainly in neuroscience, and I think in much of medicine, we're able to generate more and more data. And often the quantities of data that we generate are beyond what an individual can or one lab can analyze. And so that's one element. And we now have I'm seeing some too many things on my screen here. That's the question of attention. And so the internet and the informatics allows us to share large amounts of data. And what we found is that, material transfer agreements and trying to patent things and all of this, it doesn't really generate very much money in general, and it generates a lot of blocks, a lot of prevention or delays in being able to collaborate. And so, we came to the conclusion, at the Neuro, and this was a grassroots thing, it was not me who decided this. I mean, I wanted us to explore it, and we did. And we ended up with in the institution a unanimous vote to adopt open science principles. Now, open science is different for there's lots of definitions of open science. So we defined what it is for us, and what it is for us is really the rapid release of all data and all biological reagents no later than at the time of the first publication. Trying to encourage collaborators to do the same. In other words, when we do material transfer agreements with other institutions, I mean, we don't have material transfer agreements. Okay, we don't do that, but we try to convince them not to ask all kinds of different things. We have created some open resources like an open biobank, open early drug discovery unit, and we make everything available. And we try to avoid having restrictive intellectual property, which usually are patents. But very important is, this is all voluntary. So one of our principles is autonomy. So if somebody really doesn't want to do this, they really want to patent something, they can, and they will not be penalized in any way, shape, or form. So that's kind of the crux of it. And we've been working with other institutions, and recently we announced that, or they announced as well, that the Hotchkiss Neuroscience Institute has also adopted open science principles very similar to the ones we have. The Douglas Hospital Research Institute, which is a psychiatric hospital in Montreal, has also adopted these. And the CAMH is very advanced in looking into this. The Moafagian in Vancouver, and the University of Western Ontario Neuroscience Group also. So it's looking like we're going to have a network of institutions in neuroscience anyhow that have adopted open science principles. Mr. Tannenbaum wanted us to focus on Canada and focus on neuroscience, which is what we've been doing. So we have been approached by other groups, general hospitals, cardiac institutes, who are interested in exploring this as well. So I think part of our role is to help others kind of explore this using our experience. So it's been an adventure. I should mention that the first open science institute on Earth was the Neuro. And until the Hotchkiss joined us, we were the only one. So it is quite new. So there's no roadmap.
Congratulations on that. We look forward to learning more about it, maybe participating across our multidisciplinary platform here at Toronto. I'm going to call on Robert Chen, who has a question.
Yep. Well, thank you very much for the excellent presentation. I think your answer on the cerebellum fits very well with what we now thinking that cerebellum is very important in mediating tremor. I just want to maybe discuss, I think what we mentioned earlier about the target for surgical treatment. So the main target is the VIM, the ventral intermediate nucleus of the thalamus, whether it's ultrasound or deep brain stimulation or lesioning. And it actually fits very well with the cerebellar hypothesis, because this is the relay nucleus of the cerebellum. The cerebellum projects the VIM and then projects to the cortex. So I think by interrupting this pathway, we seem to have a significant reduction in tremor. So I think that fits very well with the what you were mentioning, the role of the cerebellum.
Okay. Well, you froze for a minute, but thanks for clarifying that. I'm not an expert in the circuitry, so that's very good. And I'm not a neurosurgeon, so I don't operate on them either.
Okay, and I think we have another question from, oh, sorry, go ahead, Rob. Yep. Oh, I think we're losing Rob there. From Jerome Title. Jerome, do you want to ask your question or do you want us to? Maybe I'll go ahead. If a FUS gain of function mutation is pathophysiologic in ALS, is a knockout a potential treatment approach to ALS?
That's a very good question. So first of all, you know, if you look at familial ALS, the FUS is responsible for only maybe 1% of familial ALS. If you look at pathology of spinal cords of all ALS, the majority of which are sporadic ALS, by far the most common pathology is TDP-43 inclusion. And it seems that TDP-43 inclusions and FUS inclusions, so in the people with FUS mutations, they get FUS inclusions that have no TDP-43, and people with TDP-43 inclusions don't have FUS inclusions. I mean, the exclusion is not perfect, but FUS is probably one of the less common, one of the rarer causes of ALS. Nonetheless, I mean, there's a treatment for SOD1, mutated SOD1, and it's probably almost as common as mutated SOD1 as a cause of ALS. So, but the problem, I think, is that a knockout would probably have to occur very early on to be able to have an effect, because we think that what happens is that you develop misfolding of the FUS, the mutated FUS, and the misfolded protein does seem to induce misfolding of other proteins. If you knock down or knock out FUS, you may slow down the process. And that, as far as I know, is actually thinking about it, there is a paper that was published using a FUS knockdown in FUS patients from New York. And it did show some benefit, but it was not a controlled study. It was one of these studies that are done because compassionate study, and the drug was developed at the New York ALS Center. Yeah, no, that's right, I remember now. So it's been done, and it had some benefit in patients who had FUS mutations. But it's a very small study, and I think there's a follow-up study that's planned.
Oh, exciting. Well,
if I could just comment, could I just comment?
Yes, go ahead, Sandra.
Because, you know, it's interesting, the TDP-43 also it can be responsible for different types of frontal temporal dementia with or without ALS. And also can be a comorbidity in Alzheimer's disease. 40% of people at autopsy, and they're usually in their 80s with Alzheimer's disease, will have this TDP-43. So I think what we're seeing is just like the amyloid and tau, there's probably some kind of reaction to some inciting event. And they're part of a sort of almost like an innate immune response that's trying to deal with something, and then it becomes pathological. And it's just interesting to sort of see this, you know, this kind of pattern unfolding in the neurodegenerative field as well.
If you had another 30 minutes, yesterday I gave a talk on ALS focused on TDP-43. So what we've done is we take spinal cords from patients with ALS. We isolate the non-soluble fraction, and these are ALS patients that have TDP-43 pathology, and some of them have frontotemporal dementia and ALS, and we can inject organoids with these with this spinal cord extract, and you can see spread of the misfolded TDP-43, which is occurring in these brain organoids. So it's actually quite fascinating. So TDP-43, and then what we see is when there's a lot of inclusions, TDP-43 is excluded from the nucleus. So normally TDP-43 is a nuclear protein, and with these inclusions, it gets sequestered outside of the nucleus. And it's thought that part of the pathophysiology of TDP-43 inclusions is the loss of function of TDP-43 in the nucleus. So it wouldn't be surprising if it did play a role in some of the other neurodegenerative diseases, and for sure frontotemporal dementia.
If you recorded it, can you make it available?
Listen, that's a good question. It was a talk I gave. Well, actually, it's a talk I gave yesterday in Hamilton, in Kingston, and I'm giving the same talk the day after tomorrow in Hamilton.
Okay, so ask them to record it and make it shareable. Thank you. That's great. That's fascinating. Thank you.
Well, on that account, Dr. Rouleau, thank you so much for tremendous presentation, exciting developments in this Open Science Institute. We look forward to collaborating, and again, congratulations on the Canada White Gardner Award.
Thank you very much.
It's really been a pleasure. Thank you.